I think we did it yesterday. I think we did it by this school.
You should.
Yeah. We're gonna get the candles out in a second.
Yeah.
Can you guys hear me? On? Yes? All right. Well, thank you for all coming, either in person or virtually, to Regina's thesis defense. I thought I would start with a picture I just learned she doesn't like. But this is the first picture I could find of her in our lab in 2018, I believe, or 2017. And so this has been a 5-year journey that started about here. And in preparing these slides, I asked Regina to send me some of her accomplishments. Some. And I got the longest list is accomplished. And so I can't go through all of these. I'm just going to highlight a few. These are just her awards, her honors, her ability to win presentation awards at local meetings. So one of the ones I want to highlight is the Research America Civic Engagement Microgrant, which was for a team that came up with a podcast. And I want to highlight this because you'll see a lot of science today. You'll see a lot of her accomplishments, but I think this is one of the best things that she's done since she's been here. It's a public outreach of science. And so I want to congratulate her and the whole team that put this together. I also want to highlight a few national and international awards. So if you go down to the second section, I don't know what you can see, the research and service recognition. She won a prestigious award from the NIH, the OSNAP Award, Outstanding Scholars and Neuroscience Award. And I think this really speaks to her impact, not just locally, but nationally and internationally. You'll see at the bottom all of her placements. She's gonna, I'm sure, deliver an amazing talk today. She always does. You can see all of the awards for those talks. The top one is actually the most impressive, and she's written it so.
Mike, can you hear me?
No, we don't want to hear you.
I know you don't, but we can see the slides advancing.
Oh my God.
Sorry.
Dr. Valdez, are your eyes open?
No.
You're just here, actually.
No, you just got to see my face.
Close your eyes.
This is so complicated. Right? So it's complicated yesterday. I mean, it really does. And
We got it, but it's presented. Should be good. Yes.
All right. Dr. Valdez, you can see that now?
Good job. Look at you.
Okay. All right, so at the bottom, under poster awards, you'll see that she nondescriptly writes best research talk by a student at Retinal Circuit Symposium. How underplayed for an international meeting of all of the experts in her field for her to win Best Talk by a Student. I received lots of emails after that talk from professors all over the world about how great it was, one of which ended up being her postdoctoral mentor. When she leaves here, she'll go and start a postdoc at UC Berkeley. So even with this long list she sent me, she really undersold all of her accomplishments. One of the things that's not on here is that she was selected to help with the grand opening of this building. You can see here with just a few of the dignitaries literally rubbing shoulders with the president of Virginia Tech, where she also delivered a wonderful presentation. And so I think, this also speaks to her ability to connect and bring science outside of the lab to a much broader audience. She's been exceptionally productive in the lab, so she's had 4 first author or co-author papers since she's been a graduate student. And she currently has kind of the manuscript that you're going to hear about today in review at eLife, and other papers also in the works. So not only has she done all of those other things I just mentioned, but she's been exceptionally productive in the lab. So I really want to talk to other graduate students now, because this is how it started, and you're going to see how it ends. But how did it go in the middle? And the middle, at least maybe the first few years, were a little rough. It took a while for her to find her science and find direction. And I oftentimes would tell her that at the second year of my PhD, I threw away every single experiment I did, and I started over because I had messed up. And it's all a process and learning. And so I actually found a picture of her during those first 2 years where she's trying to find the right path forward. And there's lots of ways that you can fail. And so here it is. You can see there's just one path forward. We protected her with a mirror helmet, and here she is navigating her doctoral work. As I said, she does make it through and does excellent job. And so here she is today at the culmination of this work. I also want to point out a few things here. This is after 2 hours of being attached to the wall. You can see kind of where we started is on the other side of this fin. We climbed up that, we crossed this little bridge, we climbed all the way up here. 2 hours later, she is the first person from my lab to make it to the top. I'm sitting there to take a picture. I took about a dozen pictures, and every one of them looks like this, right? And so you can see she's hunched over. She's giving a death grip to her leash, which is attaching her to the wall. And I, for the life of me, couldn't get her to relax and kind of put her hands up and be like, I got here, and celebrate the success. And so this is the best picture I got from that day. Now I want to say a few things, a few other things I learned about Reshma. So you saw all of these pictures, but this is not the face of Reshma, the left. This is, I like to call it
Why?
the what the hell is happening face, which happens almost every day. This is a very common look for Reshma in the lab. It's oftentimes followed by this face, which is I work with a bunch of clowns, right? And so these are the sorts of things that I'm going to remember Reshma for. But of course, she is one of the clowns too. So she fits right in with the rest of us. Other things that I have learned about Reshma that I think are worth sharing. For 4 years, she has had fake dog food on her desk. And every day I come into lab, I ask her why. And it's come to my, she never tells me, but it's clear that it's kind of like the thing that makes her comfortable. And so yesterday we had a thesis defense, many of you were here. It was a perfectly good thesis defense, but I got all sorts of texts from students because it was taking longer than they thought. And so I thought that the anxiety level might be high. And so I actually brought those for you today so that you feel comfortable in your safe space while you're presenting.
We all talk about it all the time.
And so, it's truly an honor to raise my hands up and get to introduce Reshma for her thesis topic.
Thank you for roasting me. Check 1, 2. Okay, great. Oh boy. Okay. Well, I was in super science mode, but that has yeah, I will have to get back to you for that, and you will see in my acknowledgements. Okay, let me hide the flowing controls, and people on Zoom can hear me. Okay. Okay. Well, hi everyone. My name is Rachna Somaya. For the past 5 years, I've been working as a doctoral student in Mike Fox's lab at Virginia Tech. And thank you to you all to make time to come to this. My family is on Zoom, so I'm super excited. Many of my friends have traveled from far to come here, and many are watching via Zoom. So I really appreciate that you all took some time on Friday morning. I know Friday mornings are tough. So thank you for making time for this. So today, what I'll do is I'll talk about this one big question that I've been interested during grad school. And the question is right here. Can the eye tell the cells in the brain where to go? And since I'm defending, the answer is yes. And today I'll show you how that exactly happens. My slides are one thing. Nope.
You might have to.
Yes. Select the slides. There you go.
Okay. Are they? I guess I'll have to use.
There you go.
Okay. Okay, so one of the questions that I find really fascinating in biology is how do you get from this stage when you're this tiny little egg, fertilized egg, from this stage to this stage where you have many specialized organs, and the organ that I'm interested in is the brain, and ultimately after some time or after some years, you will have this specialized circuit in the brain where you have billions and billions of cells talking to each other, sending these electrical signals so that you can have crosstalk in the brain. So how does this happen, and what exactly does this happen? What exactly happens at this stage, this question mark stage? And this is a huge process, and I cannot possibly talk about everything. So what I'm going to do is I'm going to take you on this journey where I first start with the process place where you recognize, and then I'll talk about what our lab studies, and then I'll transition into what I specifically study. So let me start at the place we all recognize. So when you're this tiny little cell, this fertilized egg, and this happens when you first conceive, when the female egg meets the male sperm, and you're just this one tiny little cell that after some weeks where cells continue to grow and multiply, and your embryo starts looking something like this. So this embryo here is called primitive streak, and it's essentially because of this linear band, which is called primitive streak. And this shape is because the cells are positioned in this weird way where embryo becomes to look something like this. So if I take this embryo and slice it to see what it looks like from inside, at this point in your embryogenesis, you'll see the 3 different germ layers of present in us. So 3 different germ layers that we usually see at this stage are ectoderm, endoderm, and mesoderm. So each of these germ layers give rise to different organs in your body. So for example, mesoderm will give rise to bones, endoderm will give rise to the stomach, but I don't care about all that organs. What I care about is the brain. So what we'll focus on is this outer layer of your germ layer, which is the ectoderm, and we'll focus on this specific area, which I'll zoom in right here. So if we zoom in this area, you'll see something called as neural plate here, and this is the part of your embryo that will give rise to brain and spinal cord. So this embryo will undergo many changes and will go through many steps, millions of steps that I'm skipping here today, but it will start changing, and at some point this neural plate will become a tube-like structure. So if you go back to the embryo to look at this in a much lower magnification, you'll see that the neural plate starts looking something like this. And if I label different things that you're seeing here, you'll start seeing 3 major divisions of brain at this point. So you have forebrain, midbrain, hindbrain. And if you're fancy pants, you can call them prosencephalon, mesencephalon, and globencephalon. So let me pull out this neural tube and let me show you what it looks like from a different angle with my primitive drawing skills. So here you'll see that these are the three same brain regions that you see within the embryo. And after years and years of development, the brain starts growing and cells starts multiplying. So our lab is really interested in understanding how do you get from this stage to that stage? And ultimately you have a brain where you have many cells talking to each other. So how does this happen? So to talk about what exactly happens at this stage, let me take this brain that you saw in like last slide and slice it. So let's slice the brain in a view called as coronal view. So this coronal view is essentially a view that's parallel to the long axis of your body. So if I'm slicing it this way, and if I take the slice and if I draw it here, this is what it usually looks like. And there's 5 different things that happen that you need to know. So the first thing that happens is you have this stem cell in your brain called neural stem cell, and this cell starts growing and multiplying. And we call these cells neurogenesis, or this process neurogenesis or gliogenesis. So neuro and glio, just so you know, are 2 different kinds of brain cells. And genesis stands for a Greek word that means being born. That's why we call it neurogenesis and gliogenesis. So the first step is enough cells are being born. After cells are born, they migrate from the place they were born to the ultimate location they will reside in the brain. So this process is called migration. So this is where they were born, and they're just immigrants, just like all of us. And they will get to the right location in the brain where they will reside. So after the cells have gotten to the right location, they have to become a specialized version of themselves. And this happens through the process of forming their own unique proteins and genes. And ultimately, what happens is they send these huge axons or nerves extending out of their cell bodies to different brain regions. And this process is essential so that you can form connections between different cell types and different cells in the brain so that you can have crosstalk between different brain regions. So this process here, together, we call it circuit formation. And I find this process of circuit formation really fascinating. And it's because if you look at different brain regions, the process of circuit formation is very asynchronous, meaning one brain region might go through this step much faster as compared to other brain regions. For example, this brain region might go through circuit formation first, and then this brain region, and then this brain region. And you might know this already. For example, the brain regions that are responsible for thoughts and feelings undergo circuit formation much later in your life versus circuit formation for brain regions that are responsible for movement. So there is this asynchronous circuit formation between different brain regions. So it's not really surprising that a brain region that's at much advanced state of circuit formation might have an effect on brain region at a much primitive stage. But this is not the whole story. Biology is not as simple as that. If you look at within one brain region, so if I focus on just this one brain region, there are different cells within one brain region, and each of the cells will go through these 5 steps that I just mentioned. So if a cell is at much advanced stage of circuit formation, it can have an influence on a cell that at much primitive stage. So I'm interested in understanding how does that happen and what's exactly the process underlying those steps. Okay, so this is a big question of circuit formation, and again, I cannot study the whole brain. So I have to pick a model, and the model that I picked was visual system or vision. So vision begins when light traverses through your eyeball. So when light first hits your eyeball, it goes through different places in your eyeball, and ultimately it will hit the inner layer of the back of your eyeball called the retina. So this is where your retina usually sits. So let me zoom in the retina and show you what retina looks like in terms of its cells within it. So within the retina, the light moves through 3 different steps, and I'm not going to label all these cells to confuse you. But I think one thing you need to know is that the light energy is present in form of photons, and the brain doesn't speak that language. So the very first step in the retina is to convert that light energy into electrical signals that your brain will ultimately understand. So once that step is done, the information about what you're seeing will enter this specific layer in the retina called retinal ganglion cells. So these cells right here sit in the final layer of the retina, and we call them RGCs or retinal ganglion cells. So retinal ganglion cells are one of the main characters of my talk, so please try to remember what they stand for. And these cells are really interesting, and it's because their cell bodies that you see here, they sit in the retina, but their axons extend out of the retina and extend out of your eyeball to enter the brain. So when these axons of RGCs, they essentially weave together, and this is what forms your optic nerve. And when they enter the brain, we refer to it as retinal inputs. So this is what I'll refer to them as for the rest of my talk. And as I was saying, they enter the brain, and when they go to the brain, they go to several places. But one of the first places that they go to, and the brain region that I'm interested in, is dorsal lateral geniculate nucleus, or DLGN. So there's many interesting things about DLGN that we don't need to know, but I think what you need to know here is this DLGN is part of thalamus, so it sits right under your cortex. So this is your cortex right here. So it sits right under the cortex. And this brain region is really important for processing of vision properly. So if something is wrong with this brain region, you won't be able to see properly. So I want to understand how circuits form in this brain region. And the way I want to understand this is at the level of molecules and cells. And to be able to do that, you have to be able to manipulate cells within any species that you're picking. And I cannot do that to humans, unfortunately. So I will use a model to understand how circuits form in LGN. So I will be using mouse as a model system to how circuits form in this brain region that's important for vision. And you might think that why should you pick mouse? Like the vision of mice is very different than vision of humans, right? And it's absolutely true. There's many differences between human vision and mouse vision. But the overall structure architecture that you're seeing here is very similar to humans, is very similar between humans and mice. So we feel comfortable in using mouse as a model system. Okay, so I'll be studying visual system, but I'll specifically be studying it in mice. Okay, so let me zoom in this brain region to show you what cells look like in this brain region and what's exactly I'll be studying. Okay, so this is the DLGN, and this is the shape of DLGN that you'll see a lot. And let me tell you why I'm showing that shape. So if you look at DLGN in mice, this is what it looks like, and this is in the coronal view. So this is the reason why I'll be using the specific schematic. So we'll take the schematic of DLGN, and we'll look at the cells that reside within the DLGN. So these are the primary cells of DLGN. And these are the cells that receive information from the retina. But you already know this, right? So I already told you that this brain region receives information from retina. But the circuits in the brain are not so simple. It's not that one neuron is just talking to one neuron. It's a lot more complicated. So just like that, the cells in this brain region also receive information from many other brain regions. And they also receive information from cells that reside within this brain region. So we call these cells interneurons. So I'm interested in understanding how does this circuitry form. And what's fascinating about the circuitry is that if you look at development and if you see what comes first in this brain region, is actually the inputs from the retina. So these cells from the retina are at much advanced stage of circuit formation versus cells such as interneurons. So I'm interested in understanding what happens if you take away these retinal inputs to these interneurons. Will something change about these interneurons? So the one big question that I've been trying to answer is, how does the first input that arrives to a region, so in this case the retinal inputs to DLGN, that arrives to DLGN might impact the development of cell types that arrive later on into this region. And one of the ways you can answer this question is by essentially getting rid of retinal inputs. So you take away this cell that's at much advanced stage of circuit formation, and then you see what's happening to interneurons. Okay, so that's exactly the approach I used. I used these mice where we did mutation in a specific gene that's responsible for the survival and maturation of optic nerve. So in these mice, there's no connections between the eye and the brain, and you can imagine these mice are going to be blind because of that. So we take these mice and we focus on interneurons specifically. So we label interneurons with green fluorescent protein. So this is why you see interneurons in the next slide with green color. And so if you look at controlled DLGN sections where there's no mutations at all, you'll see that there's a lot of interneurons, right? Everything looks great. But if you look at our MAT5 mutant mice where the connections between the eye and the brain are not present, there's significantly fewer interneurons as compared to controls, right? So something about retinal inputs or something about the information from the eye is important for interneurons to be present in this red region. So essentially the big takeaway is that the presence of thalamic interneurons is dependent on retinal input. So now we take this finding and now we try to understand what's exactly happening to these interneurons if you don't have information coming from the eyeball. Are they just dying? Are they just dying, or are they present but they just don't get to the right place? Do they just not migrate to LGN? So to understand which one's the case, we took one hypothesis at a time and started exploring it. So first question we wanted to answer was, what if the problem is cell death? What if in the absence of retinal inputs, these interneurons just die and cannot survive? So to answer this question, we labeled interneurons along with labeling for a marker that labels dead cells. So the dead cells here will be labeled by cleaved caspase 3, which is present in magenta here, and interneurons are present in green. And again, this is the LGN section. In control, you don't see anything happening, right? But if you look into mutant mice where there's fewer interneurons, I want you to pay attention to the fact that there's not a single cell that's positive for both interneurons and the dead marker signal, which is the cleaved caspase 3, suggesting that the problem is not cell death. So they are not dying in the absence of retinal inputs. But what if the problem is migration, right? That can also be the case. So to figure out that they are not getting to LGN in the absence of retinal inputs, what we did was we focused on regions neighboring LGN. So this is again the coronal section, and this is DLGN. So here you're looking at this region BB. It stands for ventrobasal complex region. You don't have to worry about remembering the name. But what you need to know here is that in mouse brain, for some reason, there's not many interneurons here. As you can see, there's none here in this section. If you're lucky, maybe you'll find one or two. So this is in controlled mice. But if you look into our MAT5 mutant mice, you'll see that there's a significant increase in the number of interneurons in ventrobasal complex region. So the region that's sitting right outside LGN. And we also show that quantification in terms of quantification. So we see this decrease in the number of interneurons in DLGN, and we see this increase in the number of interneurons right outside the DLGN. So I'm not showing you all the results here because of lack of time, but just so you know that we also confirmed that the identity of these interneurons is similar to the identity of interneurons right here. So we feel confident in saying that these interneurons were meant to be here. So it seems like in the absence of retinal inputs, it's specifically the migration of interneurons that's affected and nothing else. Something is happening with the migration of interneurons. Okay, so just as a side note, you'll see that most of my findings in schematic are focused on DLGN, but everything that I'm showing you, all the results here, we also saw them in VLGN. So VLGN, it stands for ventral lateral geniculate nucleus. It sits right under DLGN, that's why it sits ventrally, so we call it ventral lateral geniculate nucleus. I just didn't want to confuse you all by showing you too many different regions. So if you have questions regarding VLGN, please let me know. So we'll focus on DLGN for the rest of the talk. Okay, so this is our big finding, right? So something about retinal inputs, this cell type that's much advanced stage of circuit formation is specifically affecting the migration of interneurons. And I think this is an important thing to understand for the rest of my talk. So I wanted to find ways for you to remember this. So I want you to think of DLGN as this conference room. Okay, so imagine this DLGN is the conference room. And the very first person that enters into this conference room is retinal inputs. So once retinal inputs have entered this conference room, then these interneurons will enter into conference room. But if the retinal inputs never enter the conference room, these interneurons cannot get in. So it seems like Mike always tells me that it seems like these retinal inputs have some donuts in their hands that these interneurons really like. So we want to understand what are those donuts. So how does this happen, and what are the molecules and cells important for this process? Okay, so I think before I show you a lot of data and bore you, I think I should talk about why. And I purposely put it here because I think this is the best place to talk about this. I think if you look at the literature of visual systems, you'll see that we know a lot about the formation of excitatory cells, so these cells right here, but we know nothing about the formation of these inhibitory cells in this brain region. And these cells are essentially really important for controlling the amount of excitation in your brain. So if you don't know anything about this cell type, you essentially have no idea how you're able to see properly. So we want to understand how exactly this process of vision happens. Okay, so during grad school, I was able to find out 3 different players that are important for this process. So what we'll do is we'll go on this journey of identifying one player at a time to figure out how exactly each player plays a role. So let's talk about the first player right here. So the first thing we wanted to know was what's exactly happening in this brain region if you don't have information coming from the retina. So what's exactly changing at the level of gene expression and at the level of transcriptome? So to answer that question, we took our MAT5 mutant mice where there's no information coming from the retina, and we microdissected LGN from control mice and compared it to LGN that was microdissected from MAT5 mutant mice. And we essentially looked at changes in gene expression. So here you're looking at a heat map, and each row corresponds to a single gene. So you can see which gene has changed its expression in MAT5 mutant mice. And what we wanted to look at genes that are specifically important for the migration process, since I was interested in migration of interneurons. So when we were doing our analysis, there was one gene that stood out, and it was FGF15 or fibroblast growth factor 15. And from this data, we saw that the expression of this gene was significantly less in the LGN of MAT5 mutant mice. So it seems like something is happening to this gene. So FGFs, the reason we call it FGF15 because it's a huge family, and there's FGF1, 2, 3, 4, 5. And this huge family is important for many things in the brain. You name it, it's important for something. So it's important for cell proliferation, cell differentiation, cell migration. And we know that from work that's been published before. So at that time, it just seemed as an ideal candidate to explore more. So the question we asked was, is FGF15 important for interneuron migration? And anytime in science you suspect a gene to be important for something, the best way, or I should say one of the most common ways to test that would be to get rid of that gene and see what happens, right? So that's exactly what we did with FGF15. So we created these mutant mice where FGF15 was absent completely. And we looked specifically at interneurons, and we were trying to understand what's happening to interneurons. So here, interneurons are labeled with a different strategy using in situ. So what you need to know about in situ is that we'll label a specific RNA that's present only in interneurons. You can visualize interneurons here, which is present in black. And you can see that in control mice, there's a lot of them. So everything looks fine in control mice, there's no gene mutation. But if you look at our FGF15 mutant mice, there's significantly fewer interneurons present in the absence of FGF15. So it seems like FGF15 is important for interneurons. And just like we confirmed in our MAT5 mutant mice, we also confirmed in our FGF15 mutant mice that it's specifically the migration of interneurons and nothing else that's affected in the absence of this specific molecule. So something's happening with the migration of interneurons. And that's our first finding. that both retinal inputs right here and FGF15 are important for interneuron migration. So now the question is, where does FGF15 come from? We know where interneurons come. We know where retinal inputs come from. We know they're coming from the eyeball. But where is FGF15 coming from? So that's our next question. And that is the second question that I'm interested in. Which cells in LGN make FGF15? Okay, so I think I have to mention that just like anything in life, even neuroscientists like to discriminate. So for the longest time, a lot of research has been focused on neurons. And that's why, to keep up with that discrimination trend, we first looked at neurons to see if neurons make FGF15. So there are two kinds of neurons in your brain. The first kind of neuron is excitatory cells, so these excitatory neurons. And here you can see they're present in green. So this is a high MAC image of DLGN. And here we have labeled for FGF15 mRNA. So the mRNA will essentially be present in the cell type that makes that gene. But here you can see that these two signals don't really overlap with each other, suggesting that excitatory neurons don't really make FGF15 in this brain region. Okay, but there's also a second kind of neuron, right? So let's finish this neuron story. So what if it's inhibitory neuron? So we label inhibitory neuron here, the neurons that are responsible for controlling inhibition. And again, you don't really see this overlap between FGF15 mRNA signal with the inhibitory neuron signal, suggesting that neurons don't make it. Okay, so now that we know it's not neurons, we have to grudgingly move to astrocytes. And we labeled astrocytes, this glial type that has been ignored for the longest time. People have started paying attention to it now, so don't worry about it. So this cell type is really important for not just passive roles in the brain, but also active roles for circuit formation. So we focused on this cell type. And here you can see that there's this in mRNA signal and the astrocytic, they're hugging. So astrocytes make FGF15 in this brain region. So specifically, canamic astrocytes make FGF15 during the time of interneuron migration. Okay, so we have been talking about this question of how does this happen, right? And before I show you the third part of my talk, which is the longest part, I think I should summarize everything that you've seen here. So there's three things you need to know. So we have seen that in the absence of retinal inputs in this specific brain region, you see a lower expression of this molecule called FGF15. So something is happening to this molecule FGF15. The second thing we learned was if you delete this molecule from mice, you compromise interneuron migration. So essentially, it seems like this FGF15 is acting as a traffic cop that gives a green signal to these interneurons to present, to get to LGN when present in this brain region. Okay, and the final thing we saw that, interestingly, not neurons, but astrocytes make FGF15 in this brain region. Okay, so these are three separate findings, right? How do we make sense out of it? How do we put all these findings together to understand what's exactly the flow of the whole process? So based on findings that you saw just right here, we came up with a model, and this is what we think is happening. So what we think is happening is that these axons of retinal ganglion cells or RGCs or retinal inputs, whatever you call them, these axons from your eyeball, when they first enter DLGN during early brain formation, they interact with local astrocytes in this brain region. And because of this interaction, FGF15 is made. That FGF15 is important for migration of interneurons into this brain region. So to indeed say that this model is accurate, you have to do lots of experiments and it will take at least 3 or 4 PhDs. So I'm not going to answer all those questions. I think I'm going to answer one of the most important outstanding questions, which is how do RGCs or these retinal inputs interact with astrocytes or talk to astrocytes for astrocytes to influence this process? So what language do they speak with each other? And essentially, what is this molecule in magenta here? Okay, so one of the interesting things about doing science is that you can read literature and you can come up with hypotheses. And one of the best things about studying visual system is that for some reason, it's been studied a lot to understand circuit formation. And because of that, we know a lot about mechanisms underlying circuit formation within this system. And one thing that usually comes up is retinal activity. So this activity from retina, and in simple language, it's the neurotransmission from your retina. So release of glutamate or release of neurotransmitters from retina into the brain has been studied to be important for so many things. And I'm just giving you a proof here that I'm not making this up, that if you don't have to worry about reading all these titles, but what you need to know is that the activity from retina has been seen to be important for many things. So we wonder, what if activity is also important for our specific question? What if this molecule right here is glutamate that gets released by RGCs and interacts with astrocytes for it to make FGF15 and influence into neuron migration? So could it be that the activity from retina is the one that influences this process? Okay, so how do we test this? So as I said, anytime you suspect something to be important, you delete that thing and see what happens, right? So we wanted to delete release of glutamate or decrease the amount of glutamate that's being released from retina. And for people who are interested in knowing in details, I'm going to walk you through it. So what we do is we take these mice where there is Cre-inducible expression of tetanus toxin. So essentially what happens is if you take these mice and you cross it to mice where Cre is expressed in the cell type that you're interested in, it will stop the release of neurotransmitters from cells that express Cre. So we took these Cal2 Cre mice in which Cre is expressed in almost all RGCs in your retina. And essentially, if you have an overlapping expression of Cre and tetanus toxin, you will have no glutamate that's being released from the retina. So we were able to obtain these mice from a collaborator. And once we started making these mice, the progenies of these mice will have no glutamate that's being released from retina, or at least decreased amount of glutamate. So once we had these mice, we took thalamic sections from these mice and quickly looked at interneurons to see what's exactly happening to these interneurons. Is anything changing about these interneurons? And to label interneurons, we'll use the same strategy here. We'll label them using their RNA molecules, so using in situ GAD1. And you can see that in control mice, everything seems okay. There's a lot of interneurons. But now if you look into our mutant mice where we have decreased amount of neurotransmission from retina, everything still seems fine. So this was really surprising because the retinal activity has been studied for so many things. And we just hoped, or I should say, at least I hoped Mike didn't, that we'll see something here, but that doesn't seem to be the case. So does retinal activity influence interneuron migration? And the answer is no. And Mike always tells me that you can publish negative results. And just don't listen to him. But it's like, it's really, it still hurts because you're hoping a lot from your dissertation and it doesn't work. So after crying for 2 days in my bathroom, we came up with an alternative hypothesis. And this alternative hypothesis revolved around another molecule. So not the neurotransmission, a released molecule from the retina. So this molecule is sonic hedgehog. So SHH, this is the abbreviation you'll see a lot. And what we think is happening is that the sonic hedgehog is being released from the retina that gets interacted with the astrocytes in LGN, and then astrocytes will be able to do everything that you're seeing here. So there are many reasons why we came up with this hypothesis, but one of the most dominant reasons we thought this would be a good hypothesis to explore was because if you look at FGF15 expression in the embryonic brain, so we are studying everything in postnatal brain after the mouse has been born. So before that, in the embryonic brain, FGF15 is expressed a lot. So we wanted to see how is FGF15 expressed during those time points. And the most dominant pathway through which this happens in the embryonic brain is through sonic hedgehog signaling pathway. So we wondered, what if it's consistent throughout the whole lifetime? What if even after the mice are gone, it's the same pathway that regulates expression of FGF15. So that's our new hypothesis. Okay, so that's our next question. Is sonic hedgehog signaling important for FGF15 expression, a molecule that's really important for migration of cells in DLGN? Okay, so before I start doing weird mutations in mice, I think I should talk about how this pathway usually works. And since this pathway has been studied a lot, we know exactly the way through which this pathway works. And usually this involves interaction between two cells. So cell 1 will make sonic hedgehog. The sonic hedgehog will undergo lots of different changes that you don't need to worry about. And once cell 1 has made sonic hedgehog, it will release it outside the cell 1. So the sonic hedgehog is present in the extracellular space. And once it's present in the extracellular space, it binds to its receptor patch 1. So this is a canonical receptor for sonic hedgehog. This molecule has strong affinity towards it, so that's why it becomes its receptor. And once this binding between sonic hedgehog and patch 1 happens, another molecule called smoothend will be activated. And then this smoothend will activate a set of transcription factors called GLI. And these transcription factors do many things, but I think what you need to know here is that they enter the nucleus and they will induce gene expression. So this transcription factor specifically induces the expression of FGF15. And this is the pathway. I've simplified it. So if you look into literature, it involves 20 different proteins. So we'll just focus on 3 proteins right here: patch1, smoothend, and GLI1. So now our question is, if we think this is the pathway through which this happens, the cell 1 has to be RGCs, right? So cell 1 has to be able to make sonic hedgehog. And cell 2 has to be astrocytes that will make the downstream components of sonic hedgehog signaling pathway. So is that the case? So that's our question. So let's first talk about, can RGCs make sonic hedgehog during the time of interneuron migration? And again, one of the ways our lab studies if something makes something is through in situ. So we'll use the same technique right here. So you're using in situ, you detect the mRNA for sonic hedgehog, and this is a high mag image of retina. And you can see that there's a lot of sonic hedgehog mRNA present in this retina section. But if you want to see the cell type that you're interested in makes the gene that you're interested in, you have to be able to label it. So we use this marker that specifically labels RGCs right here, CalQ. And you can clearly see that there's an overlap between the sonic hedgehog expression and CalQ expression, suggesting that RGCs do indeed make sonic hedgehog. So I was doing this experiment around 2019, and around the same time, another paper was published that showed that RGCs not only make sonic hedgehog, but they also release it in the brain. So this gave us a good start. And now we wanted to know, can astrocytes, do they have the molecular machinery to respond to sonic hedgehog? And the first way I answered this question was looking at sequencing data. So here you look at RNA sequencing data where we have seen which RNA molecules are expressed in DLGN. And these are the 3 molecules that I talked about: GLI1, patch1, and smoothend. And as you can see, all 3 molecules are indeed expressed in DLGN. So we see the expression, but that's not our hypothesis, right? Our hypothesis doesn't stop there. We want to see if astrocytes make these molecules. So again, to understand what cell type makes it, we did in situ. And here you're looking at a high mag image of DLGN now. So this is patch1 expression, and you can clearly see that patch1 is indeed expressed. But if you co-stain this with a marker for astrocytes, you can clearly see that there's an overlap between the astrocytic signal and patch1 signal, suggesting that thalamic astrocytes do indeed have the machinery to respond to sonic hedgehog that's being released from the retina. Okay, so I'm just showing you results for patch1. We have done this through several different ways, and we have confirmed that indeed most of the molecules in sonic hedgehog signaling pathways are expressed. biothalamic astrocytes. But let me give you just one more example to show you how we do it using another technique. So here you will see cross sections from a mouse where GLI1 expressing cells were tagged with a fluorophore. So GLI1, just as a reminder, it's the transcription factor that's expressed in sonic Hedgehog signaling pathway. And you can clearly see that the GLI1 expressing cells are indeed present in DLGN and DLGN both that I've been ignoring all this time. But you can see that, yes, if you look at it at a better magnification, the GLI1 expressing cells are present, but if you stain it with another marker for astrocytes, so for people who cannot read this is S100. It's also one of the most commonly used markers for astrocytes. You see that there's indeed an overlap between these two signals, suggesting that thalamic astrocytes make sonic Hedgehog signaling components. Also, I didn't honestly have to do this staining. For people who study astrocytes, they can clearly tell the morphology of the cells looks like an astrocyte. But it was a good confirmation. Okay, so are sonic Hedgehog signaling components expressed in the system? Yes, they are expressed. But that's not our question, right? That's not the exciting part. The exciting part is answering the question of, are sonic Hedgehog signaling components necessary for circuit formation in this brain region? And the way we are answering this question is by looking at FGF15 expression and interneuron migration. So how do we test this? So we were trying to come up with different strategies. I think the best strategy was to delete sonic Hedgehog from retina. And then you look into the brain. So you're deleting something from your retina, and then you look into LGN to see if anything is compromised at all. So before we do this mutation, I think you have to step back and you have to answer, is this really a good strategy? Because many times these crossing of different lines doesn't really work. So we wanted to know if this crossing of sonic Hedgehog flox line to Cal2 line is indeed a good strategy. And the reason why it's an important question to ask here is because if you look at how complex the retina is, it always surprises me. So this paper right here in 2018 did single cell sequencing in retina to identify different types of RGCs in retina, in mouse retina. And you can clearly see that there's more than 40 different types of RGCs. So if we want our pre-lock strategy to work, we have to make sure that both sonic Hedgehog and Cal2 are indeed expressed in the same type of RGCs. Because if there's no overlapping expression, the mutation is not going to happen. So is that the case? So to see if that's the case, what I did was I took single cell sequencing data from this paper and reanalyzed it for my question. And this is what it looks like. So this is a GG plot. Let me walk you through it. So on x-axis, you're seeing expression of sonic Hedgehog, so this molecule right here. On y-axis, you're seeing expression of Cal2, which is the marker of this cell type. And each colored ring corresponds to one type of RGC. So there's many takeaways from this plot, but I think the biggest takeaway right now is that every single cell that makes sonic Hedgehog also makes a lot of Cal2. So it seems like there is an overlapping expression of sonic Hedgehog and Cal2, suggesting that this strategy would at least work in principle. Okay, so is it really working? And to see if the strategy is indeed working, we did a simple qPCR experiment where we looked at changes in the expression of sonic Hedgehog in the retina. So this here you're seeing expression of sonic Hedgehog in the control retinas. And you can clearly see that in our mutant retinas, there's lower expression of sonic Hedgehog. So we are able to do this mutation in the retina specifically. And now we can look into DLGN and in the brain to see what's happening. Okay, so the first thing I wanted to know was, is something happening to FGF15? Are you seeing anything going wrong with FGF15? And again, I'll be doing your in situ. And I think I should mention here that I'm doing in situ over immunohistochemistry is because for some reason, it's really difficult to have a good antibody for FGF15. We have many hypotheses why we don't have good antibody for it. One of the reasons is that it's a released molecule in the brain, so it's difficult for the antibody to detect that. The other reason why we think there's no good antibody is because if you look, even if you look into control brain where there's no mutation, the expression of FGF15 is very low. So it's just a low expressed molecule, which is hard to detect via antibodies. So we overcame that issue by making a riboprobe. And we stain for FGF15 in our control DLGN sections and our mutant DLGN sections. And again, here the black dot corresponds to FGF15 mRNA. And you can clearly see that there's a lot of it, but if you look into mutant DLGN, there's this dramatic decrease in FGF15 expression. And we also show that quantitatively as well. Okay, so it seems like this molecule from retina is important for FGF15 expression. But we know that FGF15 is important for interneuron migration. So is that the case? So is something happening to interneurons in the absence of sonic hijab from the retina? Okay, so to look at retinas and to keep things consistent and not to complicate everything, we again did in situ to detect GAD1, a molecule that's present in the retina. And you can clearly see that there's a lot of it in control mice. But if you look into our mutant DLGN, where you don't have this information coming from your eyeball, you see significantly fewer interneurons in this brain region. So this experiment that you're seeing here was done at a mouse that was maybe 7 days old, and that's when they finished the migration of interneurons. So we wanted to see, what if is this problem consistent, or what if this is just a delayed process? What if we looked at older mice where there's matured circuits, will you still see a compromise in interneuron migration? Is that the case? Okay, so I repeated the same experiment, and this time I did it in mice that are much older and they have much matured circuit. And you can see that even when the mice are a few months old, you see this like significantly fewer interneurons into this brain region if you take away sonic hedgehog from retina. So it seems like this signaling pathway is important for FGF15 expression and also interneuron migration. Okay, I am almost done, and this is my final slide. So I think I want to conclude it in a few different ways. But I think let me summarize everything that you've seen here. We have made the schematic so that it brings everything together. Because I know I talked a lot about different molecules and different cell types. And I think this picture summarizes everything. So even if you zoned out, if you come back here, I think it will make sense. So what we think is happening is that this axons from your eyeball, retinal ganglion cells, when they send their axons to DLGN, they interact with local astrocytes in this brain region. And because of this interaction, FGF15 is made, and this FGF15 is important for interneuron migration. So it seems like there's this crosstalk between 3 completely different cell types in the brain, an inhibitory cell, an excitatory cell, and an astrocyte, which becomes ultimately important for circuit formation in this brain region. So why is this cool and why should you care? I think there are many reasons why you should care, but in the context of visual system, but I'm not going to talk about it in the context of visual system because I think that's boring for people who don't study visual system. What I'm going to talk about is 3 reasons why this is important in the broader context. The first way I want to talk about this is how this project shows us that there's an interplay between neuronal activity in the brain and molecules. So during early brain formation, if you see what's important, at some time you'll see that activity, this neurotransmission from retina is important, and later times you'll see that molecules are important. For this project, we saw that activity is not important for circuit formation for interneuron migration, but it seems like this released molecule specifically is important. So it really fascinates me to think about how does the brain decide what it needs in a specific context. And I think that's an outstanding question that I hope to answer one day. Okay, the second reason why I think you should care is because I think this project forces you to think about cell-to-cell communication. So for some reason, the migration of interneurons is dependent on this crosstalk between excitatory cells and astrocytes. it's just really fascinating to me why interneurons are not programmed within themselves to get to the right place. So why does the brain have to build this complex system for the interneurons to get to the right place? Why do you need this complexity? And I think anytime you ask the question of why does something happen, the best way to answer that question, in my opinion, is to look at evolution and to answer the question of, is there an evolutionary origin to cell signaling? And I think it depends on who you talk to. So if you talk to this guy, does anyone recognize who this is? Okay.
Never heard of them.
This is Charles Darwin. So if you talk to this guy and if you read his On His Origin Species book, which is this huge book that I tried to finish, but I think I can never finish. But if you read this book, according to him, the argument is it was the successive mutations that forced the cells to interact with each other. It was not the choice they made. But there's this long history of evolutionary biologists that argue against Darwin. And their argument is that the cells chose to interact with each other, and complex cell-to-cell signaling was just a consequence of it. So I don't know whose side you're on. I have picked a side, but I'm not going to say whose side I'm on. But I think no matter whose side you pick, we cannot argue that this complex cell-to-cell signaling gives us an advantage. It makes our circuitry more complex, you have more diverse functions, and you're able to do many more things. And keeping up with my evolutionary theme, I think another reason why this is important is because I think it forces you to ask the question of, you can see, why do we need Rachna Dev and Somaya? That's not, why do we need astrocytes? I don't know how to move this. Oh, I know how to move this. Okay, so why do we need astrocytes? You know, these two cells, retinal ganglion cells and interneurons, they're both neurons. They could easily just talk to each other, speak a language that they understand. Why do we need this middleman that's very different from each other? And again, I think going back to evolution, does everything that possess nervous system, does it need astrocytes? And the answer is no. So if you look at animals with diffuse nervous system, for example, jellyfish, they do have supportive cells, but they're not really astrocytes, and they don't need those supportive cells to have their circuits being formed. But as circuits become more and more complicated or diversified and become centralized, you'll see that these cells called astrocytes are starting to be present, and they are more involved in these processes. So we saw this here in mouse. My guess is if we look at this in humans, this is going to be much more complicated. And I think that just speaks to how the evolution gives us an advantage by having many more diverse cell types, a much complex circuitry via these like signaling mechanisms. Okay, so this is the part that I will keep myself together to do it. I don't think this is a time for my swan song, and I think I wouldn't have gotten here without this person right here. I think you cannot, if someone tells you they did their PhD by themselves, I think it's a lie, they're not being honest. And I think someone who is really the most important person, I would say, I'll start with the most important and end with the most important one. And this is my mentor, Mike. And I don't know, Mike, if you remember this, in my qualifying exams, so qualifying exams are exams that you do right after your first year. And at that time, Mike asked me to draw the shape of DLGN, the shape that you saw a lot, and I couldn't do it because I didn't know at that time. So at that time, that was the level of knowledge I had, and he took a chance on me. And I distinctly remember I was competing with many people to get the spot. I thought I was giving my best, but I knew my best was nowhere close to what's actually best. So I really appreciate you taking a chance on me, for everything that you've done to be patient with me while I've done so much troubleshooting. But I think this picture doesn't capture his true personality. I think these are the two pictures that capture his best personality. So I give him a lot of hard time for being busy, especially now. But even when he's busy, he takes time to celebrate little achievements by his students. So by bringing cupcakes when he knows everyone is health conscious in the lab, or he'll leave notes, let's say happy birthday, even when he has back-to-back meetings. So it just blows my mind how he's able to do science and also make time for his trainees. I wish I'll be able to do that when I become a PI. And that's also my hint to tell you all to recruit me to FPRI as a professor. I think this picture also doesn't capture his true personality. He is not just a mentor to us in science, but also a mentor to us outside of science. And for some reason, he tends to pick people who don't know how to swim, don't know how to play mini golf, and just basically know how to do science. So in this video, he's trying to get everyone together and teach them how to mini golf. And I'm going to show you how patient he is with all of us. And even when he knows that we are giving him a hard time, he always has a smile on his face and wears t-shirts that says, life is good. And if this can play. Because I want to remember. The Jimmy is going Jimmy. I had to show our lab dynamic. Yeah. Good. Okay. Oh, thank you. And we couldn't do it. It's that complicated. Thank you, Mike, for being the best boss I could have asked for. I know you hate cheesy stuff, but I had to make it a little cheesy. And I hope we continue to collaborate after I move to Montla's lab. And I think I have said this publicly before, and I'm probably stupid to say this right now when my committee members are present, but I think Fox Lab is the best lab at FPRI. And I don't say that because of Mike, but I also say because of everyone who works in Fox Lab. And this includes all the people who have been with me throughout these 5 years. Ubara, Jimmy, Caitlin, Abuzar, Yanping, Gabby, Alex, Alicia, my mentees, just everyone has been wonderful. And somehow they also know how to do good science, but also collaborate with each other. And I couldn't have asked for a better lab to do my grad school. So thank you to each one of you to deal with me, I guess. Thank you to my committee members. I think I have gone to each one of you at some point for different reasons to get suggestions on how to improve. And Shannon has helped me a lot. Even times when I have not interacted with you, I have looked up your profiles to see what I can do to get to your stage. Shannon, Mike, Stephanie, Greg, all 4 of you have helped me a lot to get here. And it was truly an honor and privilege to learn from each one of you. I also want to thank our collaborator, Dr. Guido. He was kind enough to host me in his laboratory for a month to teach me electrophysiology, and I still don't know how to do it, but the fact that he knew that he could take a chance on me and pay for my trip was really kind of him, and I really appreciate everything you all have done for me. I also want to thank TVMH program. I think, I mean, like I said, the best lab, but also the best program. And I was showing this to my friend yesterday. She told me it looks like these people are dead because of the and none of these people are dead. They're alive. They just don't work here anymore. But these people were very important towards the initial process of my grad school. So I'm thankful to them, and I'm also thankful to everyone who works here right now. I think you all have created a wonderful program. I've gone to each one of you to complain something about grad school, and each one of you have changed it immediately. So thank you for making this a best experience. I just say, I think to many of these people right here, they were just doing their jobs, but you have no idea how incredible and integral your role was to my PhD. And that's why I call it the unsung hero. Each one of you has played a huge amount of role for me to get here. Mike, you have built a wonderful community, a collaborative community. And for some reason, you decided to pick me to speak for the opening of the new building. And I had the biggest imposter syndrome. I was like, why am I being picked here? I'm like the least successful person, and I'm going to get out of here as quickly as I can before they know I'm a fraud. And I'm thankful to each one of you to make me feel included. Everyone, the IT department, people who took care of my monies, communications department, HR, Hal, Irvin, and Brennan, people who get money to the institute, everyone in the admin team, everyone who ran all the operations. Thank you to each one of you. I think people say money is not everything. Money is something, trust me, and you cannot do much without the money. I was able to do a lot of these projects because we had fundings from NIH and UK. But I have complained during my grad school a lot how I was competing in this intensely competitive environment with not the same amount of resources that many other people had, because I was not eligible for many fellowships, but many people heard that. And I was able to obtain a competitive fellowship last year through generosity of Ray Gaskins, and I couldn't be more thankful for that. So thank you. I don't want to do this, but I mean, many of my friends are here, so thank you to all for taking time out for this. I really couldn't have done this without any of you. As an immigrant, your whole family is not next to you, so your friends become your family. So I'm really glad to have met each one of you. I mean, I have tried to put everyone's pictures I can, and just thank you for being my friend. I know I could be a lot, and it's just the wonderful people who are my best friends, people who are like my family. It's just each one of you has played an integral part for me to get here. So thank you so much. And I strongly believe if you can work or live with your friends, they're your better friends. And I was able to work with some of my friends, and we were able to create this wonderful podcast. And I know this is much smaller picture than what Paris put yesterday, but I promise I'm equally thankful to each one of you. And I hope we can continue this podcast once we move from FPRI. Okay, so I think my family is on Zoom. So I really don't think for me to get here, I don't think I was the person who got me here. I think it was my family who got me here. And even now, they're just working and working. And I have only gotten this far in life because of these two people right here. So this was my dad and my mom. This was one of the first pictures. I think I was maybe 1 or 2. I don't remember this. And even when we didn't have any money at all, I knew my dad worked hard to celebrate it and to send me here. And I have never seen someone work so hard as he has. And my mom is the glue that keeps our family together, because without her, we'd be just a bunch of workaholics. And I've never seen someone have so much determination and drive to thrive in any situation life puts at you. And I'm only here because of these two people right here. So I should say this is not how we look right now at all. Things have changed. They don't look like that anymore. And this is what we look like now. And this was the picture that I took when I first came to the US. This was when I first was coming to the US. And little did I know my life was going to completely change. At that time, I did tell my parents that I would come back to India, trust me, and I had lied. And I don't think I'm going to come back, mom and dad. I would like to be here, but this is not the way I tell you this. This is to show that they've done everything they can for me to be here. And my dad always jokes that it was because of me he became from this to this. And I'd like to clarify in front of many scientists that I was not responsible for this process. It was normal aging process. I might have fussed in this process, but I am thankful to both of you. I think you all got the best sibling to work with. My brother, who is not so smart, he's going to get me so much to do this, but this was the best way to get back to him. This was him acting that he was studying when in fact he was just watching YouTube videos. He's 7 years younger than I am, but he doesn't realize in so many ways he's helped me to get here. So thank you to all my family. Okay, I picked May 6th for my defense, not because I like May 6th. I picked May 6th because last year on this date, I lost my grandfather and I couldn't go home because of COVID. And I hope wherever he is, I hope he is proud of me and he is resting in peace. And I haven't gone home in 4 years, so I hope to go home and I can't wait to go home. So thank you to listen to me to that sappy slide. And now it's the time for you to prove that you didn't fall asleep by asking questions.
All of you did this.
Hey, Rashna, I
Yeah.
have a question. It was really interesting how astrocytes respond to sonic hedgehog in the DLGN. Do you know whether astrocytes in other parts of the brain respond to hedgehog in the same way, or is this a specialized type of astrocyte?
Yeah, that is a good question. So for the people on Zoom, the question is, for the people on Zoom, the question is, are astrocytes on any other brain region or any other context also responding to sonic hedgehog, or is this a specialized circuit, right? Okay, so I think the answer is yes. The short answer is yes. We know, especially this for injury context, that neurons will release sonic hedgehog and astrocytes will respond to it. And we also know this in the context of synaptogenesis, especially in the cortex. But this is the first time anyone has shown it in thalamus. So it's not that it's novel in terms of mechanism, but it's novel in terms of where it's happening.
And in terms of recruiting interneurons or recruiting
This
cells
is-
migrating.
Yeah, this is the first time. Yeah, so people who have shown this have not shown this for migration, but they have shown this for synaptogenesis.
I have a small question. So you mainly focus on the upstream of the FGF. Process is decreased and affected into neuromigrations. We, of course, our lab mainly focus on visual system, which are retinal ganglion cells activity. And you think about the other visual related part, or like visual cortex, they also can project to DLG. Is it possible they also involve this interneural migration to DLG or DLG,
So
especially DLG?
yeah, I think, is it possible in terms of have I seen it? I have not tested that hypothesis, but if I have to speculate, I would think no, because the cortical inputs come around the same time as interneurons. So it's not like they get there before interneurons for them to have an influence. So my guess would be for at least DLGN that might not be the case. In DLGN, I'm not sure. But yes, but as of now, I would say for DLGN, I don't think that might be the case.
Because the reason I asked this, as you crossed with the carotene not only expressed in retina, it's also mainly a lot of expressed in cortex, especially visual cortex. That's why I focused on.
Yeah, I mean,
I need to
that's,
check with the sonic hedgehog, is it expressed in visual cortex or not?
yeah.
When you delete it, maybe also delete it in visual cortex part, then make it cannot regulate to the FGF.
Yeah, I mean, yeah, definitely. I think overlap seeing the question is, I should have repeated the question. The question is, do cortical inputs in DLGN also have an influence in this process of interneuron migration, along with sonic hedgehog coming in from the retina? And yeah, I think the first step would be to see if there is indeed an overlapping expression of sonic hedgehog and TAB2 in cortex, to see if my model is also having that has that confounding effect because of that. So that would be a good experiment to do afterwards. Maybe you can do it after I leave.
I understand that sonic hedgehog is basically like a sledgehammer to the developmental process, but is there clinical data in human patients, especially in children, that they have visual defects when they have mutations in the sonic hedgehog pathway?
Yeah.
And is there any evidence for human data to support what you found in your mouse system?
So yeah, I mean, that is an interesting question because it puts everything in clinical context, right? So mutations in sonic hedgehog are extremely lethal in human beings. There's so many things happening, intellectual disabilities, like problems with their facial structures, that I wouldn't be surprised if something is also wrong with their visual system. no one has specifically looked at interneurons, but I think it could very well be the case. The fact is that people are so focused in those humans in like intellectual disabilities that we're still not getting to the place where we can look at vision. But yes, it's definitely a possibility. Anything on Zoom? Or is no? Ubara has his hands up, he wants to speak. Okay.
I don't have my hand up. Can you hear me if I speak?
Yeah, we can hear you.
Oh, okay. Well, I took so much time typing this. Okay, fantastic talk. Congratulations. I was wondering if when you crossed the tetanus animal to CALP2, right? You crossed it to CALP2 or VGLUT2.
CALP2.
Yeah, so when you did that cross, I know you didn't see a phenotype, right, in terms of the interneuron count in LGN. But I'm wondering, like, did you see any other like obvious phenotype to do with like other places that CALP2 is expressed, which is all over the place, like, you know, locomotion or anxiety or even vision, since you are silencing, you know, activity in retinal ganglion cells. And like Jimmy said, I think it was Jimmy, like Jimmy said that, you know, cortical interneurons, a lot of them express CALP2 as well, cerebellum. So I'm just wondering, like, did you see any obvious phenotype? I know you didn't like look for it, but was there anything weird about the animals?
No, there was, yeah, isn't that surprising? Like you're removing neurotransmission from such an important cell type, which is not just present in retina, but it's also present in cerebellum, for example. Yeah, we don't see anything. I don't know how to explain it. I mean, does that mean that neurotransmission is not so critical for showing us gross phenotypes? And I would argue right now, yes, it seems like it. So we didn't see anything. There were times when some of the mice appeared smaller, but it was not so smaller that it stood out. It was only when I was trying to look for something. So it doesn't seem like that is the case. And even when they're able to breathe fine, everything's okay with them. So I don't know what is happening. So I think it goes back to the question of how important is neurotransmission early on. And as of now, I think it doesn't seem like that much. It's a very really risky line to say that I'm going to say it.
Okay, can I ask another one?
Yeah, go ahead.
Did you consider at all like delivering sonic hedgehog in a mutant animal, delivering it to the LGN and seeing what happens?
You mean doing injections?
Yeah, like like a recombinant protein or something,
Oh,
and then seeing if you can rescue. I mean, you know, essentially.
Yeah, no, that is, yeah, I think that would be the solid experiment that will solidify everything, right? Yeah, I haven't done that. It's the next thing to do, next right thing to do. So to take one of our mutant mice, whatever it is, like FGF15 or not FGF15, MAT5 mutant mice or sonic hedgehog mutant mice, and do DLGN injections into it, and to see if we can get interneurons. I think we could also inject sonic hedgehog in places outside of DLGN where there are not many interneurons to see if we get interneurons there. So yeah, I think that would be the next right thing to do, but I haven't done it yet.
But the astrocytes outside the LGN don't express FGF15, right? Or do they?
That's a good point. So I think what I should be injecting is FGF15 outside of LGN.
Yeah,
yeah.
Well, have fun with that experiment, Jimmy.
He's ignoring us. Yes.
I welcome you back to do it.
Yeah, this is awesome. I can't put into words how much I learned in the past hour, so I really appreciate the way you delivered it. My question, I guess, is a little bit of a fun one. Obviously, you've learned a ton about a lot of different areas, the eyes, brains, whatever. What was something that you learned about yourself through the process?
Like with the grad school?
Yeah.
Yeah, I mean, that is I think what I learned was I don't know how to ask for help. When things go wrong, I will struggle for years, and then I will go to Mike's office in Fry for after some years. So I think, yeah, I think I learned that in my postdoc I shouldn't be afraid to ask for help. And I say that to even future grad students. I was too prideful to ask for help. And I think I'm still there, but I am changing. But yeah, but that is an important question. Nothing else? Oh, I'm PhD. No other questions in Zoom? Okay.
I mean, it's a meeting not done yet, you're not PhD yet.
What?
Although it's sure.
No. You don't say that Mike will fail me just because you said it.
Only after 2 hours decide. I'm kidding.
Good luck to you all for making